Let's discuss on CRISPR-Cas9 gene editing, an introduction to revolutionary gene editing technology. This technology has revolutionized the genetic engineering, and with this technique, now it is possible to modify, delete, insert, or activate or inactivate any of the gene. So, what is CRISPR-Cas9? CRISPR, CRISPR, that stands for clustered, regularly interspaced, short palindromic repeats. What is Cas9? It is the CRISPR associated protein 9. And this Cas9 gene, it codes for a number of proteins, which is what the nucleases, helicase, and polymerase and polynucleotide binding protein activity. And this protein the protein derived from Cas9 gene are the enzymes that act like a pair of molecular scissors capable of cutting the DNA strands. But the Cas9 does not just cut anywhere in the genome. There are certain specific sequences present over the target DNA, which are known as spam sequences or protospacer adjacent motifs. So they serve as a tabs, and at these sites, this Cas9, near to these sites, the Cas9 cuts the target DNA sequences. Now, a genome editing tool that allows researchers to alter the DNA sequences. So this CRISPR-Cas9 is this what we call as genome editing tool. This CRISPR system is very much advanced in the bacteria and other lower microorganism. It acts like a bacterial immune system. Acquired immune system stores fragment of viral DNA in CRISPR sequences. Cas9 uses these sequences to recognize and cut future viral DNAs. Then what are the components of CRISPR-Cas9? It has got a guide RNA or gRNA. It matches and binds to the target DNA sequences. Then Cas9 enzymes. These cut the DNA at targeted location. Already told you near the PAM sequences, it cuts at the specific sites onto the target DNA. Then target DNA, that is that sequence of the DNA which is to be modified. So these are the various component of the CRISPR-Cas9. What is the mechanism of action? I will show you a pictographic of how the mechanism works. But let's see here. The guide RNA binds to complementary DNA sequences. Then Cas9 cuts both strands of DNA at these sites. Then cell attempts to repair DNA breaks via two procedures. One is the non-homologous end joining. Second is the homology directed repair. So non-homologous end joining, it's from the cell side and it may lead to mutations. Because here the lost nucleotides, they cannot be filled in the gaps. Then homology directed repairs, it uses a template of nucleotides for accurate repair and filling the gaps. Applications of CRISPR technique, it's in correcting the genetic mutations via gene therapy, developing disease resistant crops, modeling the human disease in the animals, studying the gene functions. What are the advantages? High precision and efficiency, then low cost compared to the older techniques. It is very versatile, applicable to many organisms. Results yielded are rapid. Then ethical considerations, potentials. It has got the potential for human germline modification. So there is a risk of creating the designer babies as well. So, and unintended consequences can be there. So, there is a need for international regulations. What can be the limitations associated with the technique? Off-target mutations are the possibility, then challenges in delivery system, then ethical and social issues are there, and requires a high technical expertise. Now, what is the summary? CRISPR-9, Cas9 is a transformative technology, no doubt. It offers unprecedented control over the genetic material, must be used with caution and responsibility. So let's see how this CRISPR as a genome editing tool, how it works in the gene therapy. So a stretch of around 20 nucleotide base pair that matches the gene of interest is generally made, and which used to be generally RNA. An RNA molecule complementary to those 20 base pair is being constructed, and the construct should be such that the nucleotide sequence is found only in the target gene. Suppose this is the target gene, so it should be complementary to this target DNA and nowhere else in the genome. So its specificity will be increased. Then the RNA and Cas9 will, these RNA and the Cas9 associated with this will identify the region of host DNA. This is the host DNA, and it has identified, and it will cut at the particular specific sequences at particular sites. Then once the DNA is cut, the repair mechanism operates. So this is the cut DNA. Now how it is to be repaired? So there are two ways to repair. One is the non-homologous way. So this non-homologous end joining mechanism will repair, but this will lead to mutations. This may cause mutation, which could disrupt a gene. So another way can be the break should be fixed with by filling the gaps with a sequence of nucleotide. Here, some known sequences of the nucleotides are being added to fill the gaps. And for this, the cell uses a short strand of DNA as template, which can be artificially supplied in the form of inserting any desired nucleotide or any gene to be inserted. So thus a corrected gene could be incorporated. So this technology is now experimentally used to correct disease, such as many genetic diseases like cystic fibrosis, Fanconi anemia. Recently, a baby was being cured who was suffering from CPS1 deficiency. So the CRISPR technique is still in experimental stage, and the CRISPR regulation is named as CRISPR interference or CRISPR activation. So this technology has revolutionized really the field of molecular biology, and with this it is possible to modify, to delete, or to insert, or to activate, or to inactivate any of the gene. So thus a troublesome gene can be inactivated by this technique, or CRISPR can be combined with the gene therapy so that modified gene can be delivered anywhere in the body. So this is the major advantages. But as I've already discussed, many disadvantages and limitations are associated with this particular technique. So, but nowadays it is held as a future therapy of genetic diseases. Lot of research is going on, and the hope is there in the forthcoming years we'll have a more advanced version of this technique, so that we'll have less limitations and more chances of cure. Thank you.